Hello everyone, this is Xiaonan Shan. So so now we are trying to do this lecture, this University of Houston ECE 6397 Advanced Imaging Technique. So this lecture will cover the optical microscope second part. So the first part we have go through this the basic concept of optical or light microscope, including this single microscope and the compound microscope, and then we introduce this infinite correct optical system, and then so we have introduced this upright and inverted microscope and the illumination system method, including the critical and the color illumination. And then so we have covered this spatial resolution, and then numerical aperture, and then also the vertical spatial resolution. So basically it's depth of view, and then oil immerse objective, and then so we and then after that so we have started to cover this basic technique, the optical microscope technique, including the dark field microscope and fixed contrast microscope, and also DIC, the differential interference microscope. So this class we are going to cover the some fluorescence-based microscope. So the fluorescence microscope is the most powerful imaging or optical imaging techniques. There's no problem about that. And so it is an optical microscope that uses fluorescence and phosphorescence instead of a reflection and or absorption to study the property of organic or inorganic substrate. So the fluorescence, the difference between fluorescence and the phosphorescence. So the fluorescence material will emit light immediately when the radiation source stops. So for example, if you see the right bottom figure, so you see this is the fluorescence. So you excite the fluorescence molecule, and then the molecule comes down to release light immediately. So this the phosphorescence material does not immediately re-emit radiations it absorbs. So it will re-emit very slowly because it has jumped to some so-called this forbidden energy state, transition state in quantum mechanics, for example. So it has in this Jablonski diagram. So when the light is going excited a molecule, and if it translate to the interstate to this triply state by this intersystem crossing, and then it will re-emit light with a much slower speed. So that's called phosphorescence. It can illuminate for up to several hours. So, for example, this eagle structure that it illuminate for a long time. Also in your watch, so sometimes you have this phosphorescence dye in your watch to show you so you can watch time even in the dark room. So again, I like to introduce some history. So the fluorescence microscope, the first has been described by this Sir George Stokes. So he's a famous guy. So everyone knows this Stokes shift, right? The Stokes shift is related to the fluorescence, and he has described this fluorescence microscope at more than 150 years ago, and then at that time he found many flora for molecule can illuminate or irradiate light with UV excitation light. And then until 1930s, so fluorochrome has been initially to use in biological research. Yes, so this guy has a famous by this Stokes shift. So Stokes shift is not only related to fluorescence but also related to the Raman shift. So the fluorescence microscope is powerful tool. So I think everyone know that. So it's more specific. You know what you are looking at because you label it, and then so you can use it for the single molecule detection or imaging, and also you can label the different target with different probes simultaneously, so you can image multiple target. So this image on the right hand side shows you the one of the upright fluorescence microscope. So for example, this is the IP fluorescence microscope. So this is light source for IP fluorescence. So basically you shine the light through a filter, whole side of an optical component, and then go through a filter, excitation filter, and then this the beam splitter or a dielectric mirror, diachronic mirror, and then you have this another filter, this on top of that. So and then so after this this filter and also this mirror or beam splitter, a certain wavelength light will go through this objective and then shine to your samples. So if your sample has this fluorescent property, so it will re-emit light with a different lower wavelength light back to your objective, and then go through this dielectric this beam splitter, and then go through this filter, and then go to your either eyepiece or go to your CCD camera. So that's the basic structure for this upright fluorescence microscope. So the first thing is important is this excitation and emission, right? So you have to excite it, and then you wait until they emit light so you can see it. So normally people use a specific band of wavelengths to excite fluorescence molecule, and then separate the much weaker emitted fluorescence from excitation light. So normally excitation is pretty strong, and then emission light is normally is weak. So the ideal case is that, so if you have a darker background, it will be better, much better. So you only see the fluorescence molecule with a really dark background. However, this excitation light normally is several hundred thousands to a million times brighter than the emitted fluorescence. So which means you have to filter out, right? You have to filter out the excitation light, even they come back by scattering or by some reflection, although it's weak, but it's still pretty strong compared with your emission light. So that's why you need to filter out those light. So that's why you need this so-called this emission filter, right? The barrier emission filter. So first of all, you have this excitation filter. So you only need certain wavelength light, right? So let's see. Right, so you need this yellow light, right, to excite a fluorescence molecule, and that's why you need, when your light source shines through this filter, before it shines through the filter, it is like a broad band light source, and then so after goes through this excitation filter, and then only certain wavelength band certain wavelength light with a certain band. So it will go through it and then reflect it on this dichromic mirror, or it's also called beam splitter. So and then they reflect back towards to your sample, goes through your objective, and then after this illumination, the excitation, it will re-emit light. So objective will collect this re-emitted light and then go back to this dichromic filter or mirror, and then go through this emission filter. So the emission filter, the goal is trying to filter out all the rest of the excitation light. So although only small part of excitation light it will going to reflect it back, but it's still pretty strong compared with emission. So that's why this emission filter will only allow the light with lower wavelengths, with this emission wavelength spectrum, will go through this your filter. So that's the principle of this emission, excitation, and emission. So this diachronic mirror, so it's relatively it is special because it will reflect certain wavelengths light, and then will allow another wavelength, lower wavelength light, longer wavelength light. Longer wavelength light will passing through. So which means normally you need to buy certain dichromatic mirror, so it will reflect your emission excitation light, and then when the emission light come back, it will allow this longer emission wavelength light will go through this dichromatic mirror. So this thing is called filter cube. So normally your microscope will have 4 cubes or maybe 6 cubes, and you can put in different wavelength filter, so you can stain your sample with different fluorescence dye. You can switch them very easily. So that's the emission and excitation part for the fluorescence microscope. And then this Stokes shift is very important for fluorescence. So basically, the vibrational energy is lost when the electron relax from excited state back to the ground state. As a result, energy lost, this emission spectrum of an excited fluorophore is usually shift to longer wavelengths or lower energy, or with lower frequency. So when compared with absorption or excitation spectrum. So this Jablonski diagram have shown that. So you have this ground state, and then this blue line means the molecule has been excited, or the electrons in the molecule has been excited to the excited state, a singly state. And then because of vibrational relaxation, so it come down to lower excitation state, and then it jump back to the ground state. So this will generate this fluorescence emission, the fluorescence light emission. So that's why this emission energy is always smaller or the lower than the excitation energy, and so therefore the wavelength will be longer because it's inversely proportional. The energy is inverse proportional to the wavelength. So that's good thing, because it can be easily to separate the emission from excitation, right? So you can separate them, because you have to, if they are in the same wavelength, and then you cannot separate these two signal, right? So you have to separate them. Otherwise, this emission is really strong. I mean, the excitation is so strong, it will kill your emission signal. So this is another diagram to show this phosphorescence. So basically, it can jump to excited triplet state, and then coming back to the ground state with much slower speed. Okay, so that's called Stokes shift. So basically, you shift to the lower energy or longer wavelengths. Okay, so for the fluorescence, there are always two parameters very important. One is called extinction coefficient. The other one is called quantum yield. So the efficiency with which a particular fluorophore absorbs a photon of excitation light is a function of molecule cross-section, and the likelihood of absorption is known as extinction coefficient. So basically, extinction coefficient is a number to describe how good the molecule will absorb light, how good it can absorb, how many percent of light it can be absorbed. So that's called extinction coefficient. So quantum yield means after the molecule absorb light, so how much of those light will translate or will be re-emit by this molecule. So that's called quantum yield. Yeah, so it denotes as a ratio between the number of quantized emitted compared with those absorbed. So that's the quantum yield. So the quantum yield normally is less than 1. So that's because of the loss of energy through some non-radiative pathway like a heat or photochemical reaction. So because of that, so so now we know this extinction coefficient and quantum yield, and related to these two parameters is fading or quenching or photo-bleaching. So that's very important, and maybe some challenge or problem for fluorescence microscope. So the fading is a general term, so which is for the reduction of fluorescence emission intensity over time. So if you keep excited the fluorescence molecule or your sample, and then this fluorescence emission intensity will decrease over time. So that's just a general term called fading. And then the quenching and the photo bleaching are subtype of this fading, or two types of fading mechanism. So first is this photo bleaching. So photo bleaching is the irreversible, the decomposition of fluorescence molecule in the excited state because of their interaction with molecule oxygen before emission. So it haven't emit the light. It has been already a photo bleach. So the people Actually, although it's a problem for the fluorescence, because over time it will getting dimmer and dimmer. For example, those figures, these three side of figures has been taken between with 2 minutes interval, right? So you can see it's getting weaker and weaker, darker and darker fluorescence, and some fluorescence is weaker than the other one, so it's getting darker faster than the other one, like a blue fluorescence in the center. You can see it's much far quicker than the red one and the blue ones. I mean the green one. So that could be a problem. However, people has using this special so-called problem, utilize this problem to develop a two new technique. One is called FRAP, one is called flip. So for the FRAP, it is the full name is called fluorescence recovery after photo bleaching. So basically, you intentionally introduce this photo bleaching. For example, you see it. Sorry. For example, you see it from this this series of image. So you can intentionally shine a really bright bleach light pulse, and then to certain location of your membrane, for example, and then it will bleach this area's fluorescence molecule. So this fluorescence molecule will embedded in this lipid bilayer, the cell membrane. So this lipid bilayer is a dynamic, like a dynamic environment. So it's always a Brownian motion is moving those molecules around, including lipid and including fluorescence molecule. So the fluorescence molecule will looks like a boat in the sea, right? It's moving around with waves, those waves, and then because you bleach them, you kill them, and then the new boat or new fluorescent molecule will diffuse back to this depleted region. So that's why the intensity will suddenly jump down because of bleaching, and then slowly recover because of diffusion process. And then so after certain time, like 60 seconds, so it just almost totally recover. So by doing that, you can study this dynamic molecular mobility or dynamic in living cells, right? You can see how fast it diffuse back. So that's very interesting and very important. So that's called a FRAP. And the flip is the full name for flip is called this fluorescence loss in photo bleaching. So this figure on the right bottom figure shows you the FRAP and the flip. The FRAP means you deplete this area, so you see the decrease and then coming back. This flip means you look at other region. So when you deplete a certain region close, right, very close to the region you are interested, so you will see the slowly decrease of fluorescence intensity. So which is caused by diffusion, right? So these regions fluorescence molecule diffuse to this depleted region, and then so that's caused this intensity decrease. So that's called flip. So both methods can be used to study its molecular dynamic, molecular mobility. So that's a really good example. So that not always a problem can be a problem. It can be you can utilize them and then to develop a new applications based on that. So that's the one really good example. Okay, so that's related to the photo bleaching, and second is quenching. Also, is highly related to this so-called this FRAP. So I will introduce next. So the bleaching, I mean the photo bleaching means you over time, right? So you illuminate light, it will speed up this oxidation process of your molecule, and the molecule will be the bleach will change to another fluorescence molecule will change their maybe a status cannot fluoresce anymore. But the quenching is different. So quenching is when you excite the molecule to the excited state. So the photo bleaching you don't have to excite. So it before you emit light, it has been bleached. Before it has been excited, it has been molecule has been bleached. And this quenching means the after molecule has been excited to the excitation state. So supposedly it should re-emit light, right? Supposedly go back to the ground state, it should be re-emit light. However, there's all kind of a method mechanism, right? Mechanism, and then it will trigger this non-radiative energy loss. For example, so the FRET is just one example. So supposedly the blue light excite this certain light, blue light excite the molecule to the excitation state, and then some relaxation, and then supposedly going back with this green color emission. However, maybe this thermal vibration, it will release this energy to this vibration mode, right? Instead of emit the green fluorescence light, it just lost non-radiative energy lost by this interaction with your phonons, right? So this thermal vibration, right? So that's one method, one problem, right? The quenching is always a problem. So the people has utilized this quenching and to design a new technique. It's called FRET. It's called fluorescence or Foster resonance. I think most people call Foster resonance energy transfer. So it's called FRET. It's based on quenching effect. So basically, instead of a thermal or vibrational quenching, so they have another fluorescence molecule which have a lower this excitation wavelength. And then if this, so the first molecule is called donor, right? So it's called donor, and second molecule is called acceptor. So the acceptor is has a much lower excitation frequency, right? Energy compared with donor. So when these two molecules getting really close, this FRET will happen. So basically you excite the first molecule, the donor molecule, and then there's some relaxation happen. And then this energy will translate to because it's getting really close, it will translate to the second, the acceptor molecule, fluorescence molecule, by this FRET effect, Foster resonance energy transfer effect. And then the second fluorescence molecule will re-emit light with their frequency, which is a red light. So if these two molecules, if the acceptor is really far away from donor, and then so this efficiency, this FRET efficiency, the energy transfer efficiency is inversely proportional to the 6th power of distance between donor and acceptor. So which means if the acceptor is getting really far away, it doesn't have to be really far away, maybe a 10 nanometer or 20 nanometer away from the donor, and then this efficiency will be really low, and then this FRET effect will not happen. Your acceptor will not emit red light. So which means if there's no acceptor really close to your donor, so your donor, if its acceptor is really far away from the donor, so donor will get excitation, and then donor will get re-emission with the green fluorescence light. And that's now if the acceptor is getting really close to your donor. So instead of a green fluorescence, the energy will translate to the acceptor fluorescence, and it will re-emit with this red fluorescence. So you see different wavelength light, re-emission light. So obviously, so it can be used to measure distance, right? So it's very clear. So that's why the people has developed a lot of application based on this FRET principle, FRET technique. For example, it has been used to measure the protein interaction. So if you have one protein, this green color, second protein fragments or peptide protein fragments, second one with green color, I mean the blue color showing here, and then you link them together with a different fluorescence molecule. One has a higher energy, I mean the lower excitation wavelength, a smaller excitation wavelength, 440. The other one with longer excitation wavelength. And then so if these two molecules bind, if they are before they bind, they are really far away. So when you shine this 440 nanometer wavelength light, so you will get 475 re-emission, right, from this fluorescence protein. And then so there's a no FRET. If they bind together, these two fluorescence molecules getting really close, and then the FRET will happen. So instead of this 475 nanometer emission light, it will get energy will translate to the transfer to the second fluorescence molecule, which will emit 527 nanometer wavelength light. It's yellowish wavelength line. So by doing that, you can know whether this binding happens, and then based on similar principle, there are so many biosensor has been developed. For example, the first one is also the similar with this example. So basically, this binding, trying to see the binding effect. And second one is this small molecule binding with this domain, sensor domain. So when you bind, they change this conformation of this protein, and then they push these two fluorophore molecule really close to generate a fret. And also other examples like after binding they change the shape, and then you get these two fluorophore really close. And then the last one is also the proteins. So initially you have a fret, and then when these proteins digest your maybe peptide, and then these two molecules will leave each other, and you will have no fret. So that's examples. So on the left and bottom also similar examples. One of them is binding test, one of them is cutting the proteolysis, and then the third one is conformation change. When the binding happens or whatever change, the conformation will change this fret. So all this application has been widely using for this the biomolecular study on the cell. So although I heard that some people told me the fret looks pretty easy, but when you do it, it's not that as easy as you think, because there are so many interactions, non-specific interactions. It's not as easy as the cartoon shows you. Okay, so next we are going to cover this single molecule imaging. So the single molecule is always interesting. It's always interesting for people, right? So people always want to measure the single molecules. So the fluorescence microscope gives us one possible method, a means to measure these single molecules by this fluorescence label, right? So the fluorescence fluorophore molecule could emit as many as 300,000 photons before it photo bleach. So assuming 20% of those photons has been collected efficiently and detected, so which means only 60,000 photons could be detected. So obviously that so you need really sensitive detector to detect these photons. Although so nowadays those detectors is really has really high efficiency, so that's not a huge problem. So people has developed this EMCCD, right? So you can use as the PMT. You can use PMT to detect it, or the photodiode, the avalanche diode photodiode to detect this small signal. Or you can also use this so-called electron multiplying CCD, the EMCCD, for those experiments. So although this nowadays the CMOS camera is catching up, I think it's pretty similar performance compared with EMCCD. So people can monitor this single molecule for many seconds or even minutes before the photo bleaching. However, the major problem is optical background noise. So those fluorescence background signals, the autofluorescence, because many components in your optical system could have this autofluorescence effect, and which will give you a pretty big background. And then on top of the background, you only have a really weak signal, so you cannot easily see those single molecules. So that's why people has found those total internal refraction, the TIR, provided a desired combination of low background and high excitation light flash. So the TIR we have discussed in our previous lecture, right? So in very much details, right? So we have known almost everything on this total internal refraction. So we have described the equations we derive the equations for the total internal refraction. So these are the two setups for those so-called this total TIRF system, right? The total internal refraction fluorescence. So it's called TIRF system. So there are two systems. On the left-hand side is the prism base. Basically, so for the total internal refraction to happen, you have to shine the light through the high refractive index material to low refractive index material. So now your sample is in the low refractive index material, which is 1.33. It's in the culture medium, maybe 1.35 culture medium. And then so you have to use something higher than that. So normally it's a glass, which is a BK7 glass 1.5. So this is one example. So you have this prism. So you focus light through this prism and shine the light to your sample. So you have a two glass, right? So you have a top glass, which you have a hold your sample upside down onto your top glass slice. And then so because of this, if your incident angle is bigger than critical angle, so you will excite this so-called this evanescence wave, it's enhanced near field on those top glass slice. On the other side, on the bottom side of this top glass slice, which you have your cell cultured on those glass surface. And then if the wavelength is matching the excitation requirement for your fluorescence molecule, so it will excite this fluorescence. And then so you have this oil immersed objective on the bottom. So you have to have a bottom glass, right? You hold your culture medium in between the glass. You hold your cell and also culture medium in between these two glass, and then you have your oil immersed objective on the bottom to collect as much light as possible, and then you can see those emit the fluorescence light from the bottom objective. So that's the one structure. And the second structure, you can see obviously it's not easy to do by the first configuration, right? You cannot change anything in between the glass, and very critical the distance between these two glass lines. And then it looks like a pretty difficult thing. And another way, so you can use this so-called turf objective or high NA objective, high numerical aperture objective, less than 1.38, right? So you shine the light through this objective. So this objective will be served as your prism. So now you have prism on the bottom, right? So you have shine the light, and then because of this binding effect by this lens, and it will shine to your sample with a certain angle. So now if you increase this laser, if you shift this laser position to the outer, close to the edge of the objective, and then this binding angle will getting bigger and bigger. So if your numerical aperture is high enough, so eventually it will reach your so-called critical angle, and then this evanescence wave will generate it on your specimen on top of this glass slice. So you have your cell culture on top, and then so you have this re-emit light will going all the direction, including down direction. And you will collect your re-emit light by the same objective back, right? So and then you have the filter, you have the excitation filter, emission filter on the bottom of this objective, and then you do everything the same except this excitation. So now you have this evanescence wave on top of this glass slice, and you have a culture cell on top, and then the entire surface, the entire space on top of your glass now it's open, so you don't need to, you can do many things on top, right? You can add your samples, you can patch clamp your cells, you can do many things. So this is obviously is much user friendly method architecture for this turf system. So that's most people using nowadays for this turf microscope. And so although one thing is that you have to use a really high numerical aperture objective. So normally it's 1.45 to 1.6 or even 1.7. So here shows you those near field or the electric field around this the interface between this glass slice and this solution or your cell. So you shine the light to the boundary at certain angle, bigger than critical angle, and then total internal refraction will happen. And when that happens, this evanescence wave will generate it exponentially, intensity exponentially decay into your solution. So this signal is called penetration depth. So we have the equation to describe that penetration depth in our previous lecture, right? So within this penetration depth, so all the fluorophore molecule, if the wavelength is matching the excitation requirement, right? So those fluorophore molecule will be excited, and then the light will be re-emitted, and then so you will collect them from the bottom. So here shows you another example to show these two structure. So one of them is based on prism, one of them is based on this high numerical aperture. So basically, you shine the light through those objective, and then the objective will bend your light to your sample, and then those turf will happen. So here is the zoom in. So how does it work? So basically, you focus your laser beam to your back focal plane or rear focal plane of your objective, which is inside your objective, and then after you go focus, it go back to a light and go through this objective front lens, and then it give you this parallel light, and then so with a certain angle. So when you change this position, this angle will shift as well. So that's the turf setup. That's how does turf setup. So the advantage for the turf is that so now you have a totally dark background, right? Because your thickness, your penetration depth is only 200 nanometer, right? So you only image within this 200 nanometer. So the background signal from the other location will be minimized. You only have the signal with this 200 nanometer range in z direction. So all the backgrounds like 1 micron away, 3 micron away, fluorophore, it doesn't get excited, and it doesn't they will not illuminate any emit any fluorescence light. So there is almost no background signal, so which can be seen from this figure very clearly. On the top two, the A and B, are those turf image, right? So you see really bright spots on the dark background, so the signal loss ratio can reach 35. So on those bottom figure is wide field epifluorescence image, so the same region. So you can still see those fluorescence molecule, but the background is relatively strong. So the signal ratio is only 1.3. So that's why those TIRF, total internal reflection fluorescence, has been widely used for the single fluorescence molecule imaging, because it generates a really good signal ratio, really dark background. And also the other application has been developed based on this TIRF fluorescence TIRF system microscope, for example, because they only image the bottom portion of the cell. So the people has used it to study those focal adhesion formation of the cell. So on those figure on the left top figures on the left hand side, that's the epifluorescence of the cell. On the right hand side is this TIRF fluorescence of the same cell. So you can see, because of TIRF only see this 200 nanometer away from surface, so mostly you see is a focal point, the contact point between cell and your substrate. So you see it's much clear background compared with those blurry flare, the background flare image from this the epifluorescence or wide field fluorescence image. And also the people has used it to study the protein dynamics, to see those how does this protein move along this philopodia of the cell when they growing on the substrate. And other things the people has used it to study, including the vesicle release and the uptake. So the vesicle release is called exocytosis, or the vesicle uptake is called endocytosis. So this a series of snapshot is taken by the this TIRF microscope. So basically, it record this acting dynamics during this the endocytosis process. And then the right hand side is another example, the full multi-spectrum or the multi-spectrum. So basically, you use green fluorescence to image this vesicle, and then the red one, right? The red one is the red one is a vesicle, right? The red one is a vesicle. Green one is this the filament, right? The actin. Right? So you can see it dynamically with those with this TIRF system. So that's TIRF fluorescence, right? The TIR fluorescence. So for the single molecule imaging. So we have introduced the three the different fluorescence microscope, the basic concept of fluorescence microscope, and also the fading, and then the flap and flip and the fret, and also this single molecule, the TIRF system. And then the next is this also based on the fluorescence, so it's called confocal microscopes. You probably heard about it as well. So The confocal microscope is also called confocal laser scanning microscope. It's a CLSM. So it's increasing. It will increase the optical resolution and contrast by using a spatial pinhole to block out of focus light. So the challenge for the traditional wide field or IP fluorescence microscope is still the same thing, right? That, for example, on this is IP or the wide field IP fluorescence image. You see those flare from the background signal. So you see a pretty bad signal noise ratio, right? It's everywhere have this green color, the background is blurry signal. So that's the problem. So the people has figured out a way trying to solve this problem. So what they did is that, so you add one, you still use fluorescence microscope, right? So you use a laser excitation laser source, and or maybe other light source, and then you add this pinhole, right? It's called light source pinhole aperture in front of your system, and then now you have this point source, and then you focus on only one location, one spot, and then on those spots, although all the light pass, so you have this fluorescence will be emitted on all those light paths, and then this red light will be focused. And so now you add a second pinhole on this in front of the detector, the pinhole aperture. So only allow the light on your focal point will go through those pinhole. All the other background light will be blocked by this pinhole, right? On the top or bottom, light goes through those because of focus, because your lens, the position, working position. So only allowed certain light coming from certain area, focal point, will go through this pinhole. Of course, you have to adjust your pinhole, trying to achieve that, and block all the other light from the upper or lower or on the upper or lower than your focal planes. So in that way, you have a much better, much less background signal. So that's the concept of this confocal microscope. So for example, on the left hand side is this IP fluorescence, on the right hand side is this confocal. You can see it's much clear contrast, right? On the top panel is this IP fluorescence wide field. On the bottom panel is this confocal. So they are imaging the same locations. You can see much clear contrast on confocal microscope. So this is the scanning-based microscope, which means you measure, right? You measure your signal 1 point by 1 point. So that's why. So you only collect the sample signal from really small region, a really small point, and then you need a really high sensitivity detector. You don't have to have a CCD camera, but you need a really high sensitivity detector. So you can use PMT, right? Or you can use this. There's a PMT, the photo multiplier detector. PMT, and also you can use this the avalanche photodiode as a detector, and then you fastly really quickly scan through your entire surface. So that's called optical sectioning, right? Optical sectioning. So you just section one layer, right? Really quickly, and then you move your focus, and then you section the second layer, right? Another layer on top of your first layer, and you can do it relatively fast. So now you can do it with maybe 1 frame or even it's like a 20 frame per second speed. Okay, so that's the confocal, right? So the confocal, why it's called confocal? So why it's called confocal is because the pinhole, the light source pinhole and the detector pinhole aperture are confocal. So that's why it's called confocal microscope. Okay, and yeah, so normally the low noise photoplier multiplier have to be used. Okay, and then so here are the different structure for those wide field or IP fluorescence, and this point scanning or the so-called confocal. Confocal, you only image those small region. So that's the 4 different structure. The IP fluorescence confocal spinning disk. So I will show you on the next slides, and also the turf. So we just show you on the previous slides. So the confocal give you much better signal. So the inventor, yes. So I always talk about the history, and so this guy is Martin Minsky. 1955. So who want to develop a microscope to image biological events in living tissue, right? So in living tissue, you have to image pretty deep, right? So you cannot just see 1 micron away from the surface. So you have to look at least a few tenths of micron. And then, so when that happens, this the background signal is pretty bad. So that's why he has described and developed this confocal microscope. So the modern confocal microscope is pretty complex, right? So you have to control the synchronization. You have to relate your signal from the PMT and to your location, right? So you have to move your scan your optical path, right? So initial confocal microscope developed by Minsky is trying to moving the sample instead of optical system. So advantage is that so you don't need to change any optical pathway, right? So in that case, optical pathway can keep steady, and the advantage is that so all the optical pathway can be optimized, right? So you don't need to change it, which means it can be stay as optimized situation, and you only move your sample. But advantage for this method, disadvantage for this method is that so you're moving your sample, right? So you have to reconstruct the image. So when you move the sample, it causes the sample is soft, it can deform your sample, and then the location could be blurry, right? You cannot to find the exactly same location, and also you move your sample, and you cannot do too much on your sample, right? So you cannot patch clamp the sample. Your patch clamp have to move your stage together with your stage, which is pretty complicated. And then that's why the modern confocal microscope is trying to scan this optical pathway instead of scan the sample. So that's the complicated control system. So you have to do the synchronization with your optical scanning system together with your the PMT tube, right? The photo multiplier tube, together with your stage or the optical scanning system, and then you can know it. So now you have nowadays people use single beam scanning or multi beam scanning. For the single beam scanning, for the scanning system, so people use this of course computer controlled mirror, and which is driven by this galvanometers. So galvanometer is an electromagnetic effect. So basically we have mentioned this effect. It's called Ampere, right? Ampere's law, right? So on the EM wave lecture, so which is pretty interesting. So people has used it to scan your mirror, use this effect. So when you're passing through a current through a wire, so you generate a magnetic field. So this magnetic field can rotate your mirror with so that's the basic principle. You can rotate your mirror. So it has been widely used for this confocal microscope and the multi-beam scanning. So I think the last slide is spinning disk, right? Spinning disk structure, spinning disk. So that's the spinning disk. So basically, you have this disk is spinning. So you have this illumination light cover the big area, and then you have this excitation, the filter and the emission filter have this dichromatic mirror and go through this spinning disk, right? So and then you only certain region light goes through those disks, and then they generate the 3 bright spots. So you can scanning 3 locations at the same time, or you can scanning 3 different wavelengths at the same time. So that's called spinning disk multi-beam scanning. Okay, so that's confocal microscope, right? And then the next one is two photon excitation microscope. So it's a relatively newer technique than confocal microscope. So the 2-photon excitation microscope is a fluorescence imaging technique that allows imaging of live tissues up to about 1 millimeter in depth. So confocal still cannot go really deep. So that's why this 2-photon excitation microscope has been developed. So even with that, so you only can do 1 millimeter. So it's still really thin, right? So if you consider your skin, maybe it's even more than 1 millimeter thick. And then the key part is that the excitation wavelength is shorter than the emission wavelength. So that's very strange, right? So the wavelength is inversely proportional to your energy. So the shorter wavelength means lower energy. So how can you excite the higher energy emission with lower energy photon, right? So it's very difficult, or it's impossible, right? So that's why it's called two photons. You have to use two excitation photon, and both of them shine onto your molecule at same time, and also both absorbed by your molecule at same time. So that's the two photon excitation, and then you get much more energy to excite your molecule. You can use shorter wavelength light to excite longer wavelengths. Sorry, so you can use longer wavelength light to excite shorter wavelength emission. So that's the two photon excitation microscope. Okay. So that's the key, right? So you use longer wavelengths, you use lower energy, you use longer wavelengths. So that's why most people use infrared excitation light. So this figure shows you the principle for the two photon. So basically, normally you use a UV light, right? Maybe a longer the higher energy light shine to your molecule, and then you get this re-emission. So now if you have, instead of 350 nanometer single photon, you have two photons, the 700 nanometer two photons. So you excite your electron into the intermediate state, and you have another photon to excite it from the intermediate state to your excited state. And then now you have two photons excited to one electron, and then this electron will emit the same blue fluorescence light from the molecule. So that's how does this two photon works. So the key is that you can use longer wavelengths to excite shorter wavelengths. So that's why most people use infrared. So the one advantage for infrared for the tissue imaging is that so it doesn't get absorbed by the tissue as much as visible light range, UV visible light. So the infrared gets much less scattering and absorption. So that's why you can get deeper penetration or deeper imaging with this two photon microscope, because you use infrared excitation light instead of UV light or maybe the visible light. So it could be a super alternative to confocal microscope. So I will explain the reason why it can be a super for that. Okay, so that's the challenge for the deeper. So we already cover for that. So the out of focus flare is a challenge. So we always have a really strong background for the deeper tissue. So the single cell you already see is really bad, right? Like those, this is maybe single cell layer. So you already see pretty bad signal, right? So those are the single cell, right? So if you can imagine if you have this a tissue, so have a lot of cells, and it can generate a big background out of focus flare. That's why the people use confocal, right? Confocal can reduce this problem by using pinhole to exclude out of focus background fluorescence. However, the drawback for the confocal is that the excitation light will produce photo bleaching, right? And the photo toxicity throughout the specimen. Even we only collect the signal from the focal point, right? So even we only collect the signal from the focal location. So all the light pathway, all the tissue on top and on the bottom, and so because we shine the excitation light through those areas, all those areas like those top and the bottom, on top of the focal plane and on the bottom of focal plane, all those regions fluorophore will be excited. So and then they will get a photo bleaching, photo bleach or phototoxicity. So which means you cannot scan your sample for too long time. So that's the problem for this confocal microscope. And that's why this two photon excitation microscope can work, will be better. So the reason for that is this two photon microscope. In order to have this happens, you have to have a really strong light. So if we go those points first, let's go through those points first. So Yeah, so those two photons will provide three-dimensional optical sectioning without absorption. I will explain later. And above and below, without those absorption above or below the plane of focus, and then it offers increasing depth penetration because of you use infrared light to excite the fluorescence. Um, Okay, so this is the key point. So because two photon excitation depends on simultaneously absorption, the resulting fluorescence emission varies with the square of the excitation intensity. So which means so the intensity decrease, and then this re-emission fluorescence emission will decrease with square of this intensity. So which means it's decreased much faster than the excitation intensity decrease. For example, this is those focal point have really strong intensity, but if it's decay along this both up and bottom region on top of focal plane on the bottom focal plane, so this intensity will decay really quickly, and then a re-emission will decay even faster because you have this quadratic term, right? The relation square relation with the excitation intensity. So which means to get this excitation right happens, you have to focus a lot of photons in a small region, and then you can get the excitation. And so which means you need first of all you need really big high intensity light. So that's why the people use this focusing this pulse laser to focus to a really small region, and then so you are not only compress on the spatial, right? So you focus to small volume, so which means you compress your light intensity to small volume, but also you compress energy in a really short time because those pulse laser they only give a really strong light in a really short pulse, so that you compress them, you improve the photon density in a really small region, really short time. And also because of this relationship to the intensity, right? The square relationship with relationship with excitation light. So which means off focus region on top or bottom, right? On top and the bottom region. So those re-emission will be really small, or absorption will be really small, because the chance for you to have two photons to hit on the same molecule will be decay really fast with the square of your excitation uh, intensity. So which means decay really fast, which means you only excited in a really small region. All the other region will not get excited. So which means it will not get a photo bleach because you never excited. It doesn't absorb light, right? It doesn't absorb those long wavelengths infrared light. So only absorb the two infrared photon at the same time with really low opportunity. So in that case, so you can focus on really small spot and only excite on those spots. You don't affect anything on the light path by this infrared fluorescence, infrared illumination. So that's the advantage. I think. So the intensity somewhere I wrote it that the intensity is like a million times higher than the single the photon excitation fluorescence. So in order to produce a significant amount of two photon absorption, the photon density must be approximately 1 million times that required to generate the same number of one photon absorption. The consequence is that the extremely high laser power is required to generate a significant two photon excited fluorophore. So this power level is easily achieved by focusing pulse laser, right? So in which the power during the peak of the pulse is high enough to excite a significant two photon excitation, while the average laser power remain fairly low. Okay, so yeah, so and then the focal point is only location that the light will the fluorescence will be excited. All the other location will not be affected. So here shows you this single photon excitation and two photon excitation photo bleaching results. So on the left hand side, the single fluorescence single photon excitation after scanning, bleaching, right? So you see all those area has been photo bleaching. So although you only see those small regions, focal plane, for the 2 photon you only bleach on those focus planes. All the other on top and bottom, it doesn't affect it. So that's how good this 2 photon excitation microscope. So because you don't have those problems, right? So, and then you don't have the pinhole. You don't need a pinhole. You don't need to block all those out of focus fluorescence flare, and you only need those focused pulse later, right? So that's the advantage for 2 photon scanning excitation microscope. Okay, so that's about this the class we're going to cover. So next one we will introduce some this superfluorescence microscope and maybe Raman and the infrared absorption based on the time available.